This is episode 258 of the Stem Cell Podcast, the Musculo-Skeletal Axis, with Dr. Olivier Pourquié. Hey, everybody, we are Daylon and Arun. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Stem Cell Podcast, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Olivier Pourquié from Harvard Medical School. He's on the podcast to talk about his research on the precursors of muscles and vertebrae, amongst other things. We've also got our usual roundup of recent highlights and stem cell news. That's coming right up.
But first, let's talk about the ISSCR annual meeting. We, of course, know that it's a globally recognized meeting for its exceptional scientific program, weaving together a broad spectrum of topics in stem cell biology, new technologies, and clinical applications to showcase the most compelling stem cell science. Visionary scientists from around the world will converge to present their latest research findings, uncovering new insights and approaches that accelerate progress. So be sure to join us for ISSCR 2024 coming up in Germany.
Man, we're already talking about the ISSCR. I can't believe it, but it piques my interest. I'm looking forward to that meeting, getting international, rocking it in Hamburg. Arun, I can't wait. But before we get there, we got like 7 or 8 or maybe 15 shows we got to do. Let's start off the roundup today with a story about Gen Tonic, the miracle elixir from the Studer lab. You know, this is a story, like the continuation of a story that's been years in the making. You know, there have been previous efforts toward this end. And this particular story has been kind of cooking in review for a very long time. And it's about maturation, right? You know, in neuro specifically, this is a Lorenz story, but in neuro, just generally speaking, the whole idea of maturation, also the heart, Arun, and not to the heart. But in neuro particularly, the idea of like cell intrinsic maturation rate during differentiation is key, because a lot of these studies in generating the neurons and particularly recapitulating the pathology of like disease and these modeling approaches, they require this long maturation time. And specifically in neurological disease and psychiatric disorders, the pathology manifests in postnatal or adult stages, and as you know, manifests as things like dysfunction in synaptic connectivity or dendritic arborization or the electrophysiological function. So all these functional elements that underlie the disease, but they don't manifest until way on, right? And a lot of the cell maturation is dictated by the environment, of course, we know as developmental biologists. But what we found over the course of the last decade, which we've probably already known forever, just intuitively, is that there's a cell intrinsic maturation rate that prevails. And it's species specific. It runs on these species specific molecular clock. And we all know humans take a long time to cook, right? So the human neurons are going to take a relatively long time relative to these other species. And this is exemplified by these transplant studies, the seminal work from the Lorenz and other groups showing that cortical neurons, for example, transplanted into the human, cortical neurons transplanted into the developing mouse brain, you got to have 9 months before they show the mature adult-like phenotype. And here, very elegantly demonstrated in these transplant studies and rescue studies, is that if you transplant mouse or pig or human midbrain dopamine neurons into the brain of Parkinsonian rats in this, you know, to induce this functional rescue, you get the rescue. For mouse, it's after 4 weeks. For pigs, it's after 3 months. And for humans, it's after 5 months. So yeah, it tracks, right? The developmental timing and intrinsic molecular clock in these species really dictates how they're going to go and how they're going to mature. But that's not good for the disease modeling field, because they want these things to manifest in a time scale that's amenable to experimentation and is practical and less expensive, right? A lot of reasons to do it. So Lorenz and other groups in studies before, I remember like almost maybe 10 years, I don't know, 6, 7 years ago, he had a story about like a tonic, so to speak, for accelerating maturation. This story was about Gen Tonic, right? And I think the interesting thing, there's a few interesting things here. It's a cocktail of these 4 factors, pretty simple. They shorten it to gin tonic, which is patented already, probably making some money. But the key here in this study to me was the way they went about it. They had this image-based, a high content image-based study, um, where they looked at like MAP2, um, immunostaining. They looked at nuclear size morphology. They looked at immunostaining for these immediate early genes, but on a massive scale here. And maybe that's where they brought Shubin in. I don't know. Shubin Cheng's also on this paper from across the street at Weill Cornell, where I am. Lorenz is across the street from us at MSK. And presumably here, in order to do the screening, or perhaps for the beta cell differentiation, and I'll get to that. Bottom line here is that they developed this tonic using this, massive high content image-based assay. They identified these factors, showed that you could get maturation, trigger like a more mature phenotype in terms of synaptic density, electrophysiology, also the transcriptomic profile. And the other key point I think here was that they showed it was outside of the neural lineage and non-neural lineages, melanocytes, pancreatic beta cells, as I alluded to. It also was able to show accelerated maturation of those, and I think that's really the key here, for me, is that this, like other studies before it, is something in terms of a tool that might be applied across the board, not just for neural and disease modeling in that space, but also this may be fundamental to accelerating the clock across different cell types, Arun, you might be interested in. And, more fundamentally, conceptually, there's probably some downstream studies there where it's like, how does this gentonic, how does it molecularly cause that acceleration? Is it like the metabolism? We've talked about on this show a few weeks back, about how metabolism really dictated that intrinsic clock. So yeah, there's a lot of unanswered questions I think this will lead up to in terms of mechanism, but fundamentally, it's an amazing tool that I think a lot of people are excited to get their hands on to try. But ironically, Arun, I mean, a lot of people probably have already tried this. The paper was submitted May 2021. It's been on bioRxiv since June 2022. I mean, almost 2 years. What's going on in Nature Biotech with the review process, Arun?
Yeah, it's definitely a long review process, and like you said, it's been on bioRxiv for a while. In fact, there are folks in my very department who have already tried this because it's been around for so long. But it's a cool study worthy of being in Nature Biotech because of that secondary application that you alluded to at the end there. You're not just talking about the maturation of the neurons, but these other cell types too. And that's really cool. I mean, most of these studies are really specifically focused on a single cell type, but anytime you can figure out a broadly applicable small molecule compound across multiple cell types, that's very powerful. And the analogy that I can make here is to something like a ROCK inhibitor, which is not just useful for survival in iPSCs, but a broad range of cell types too. Maybe they found something similar with their gentonic cocktail. I like the name. Um, the other thing to allude to is, obviously the expertise of Shubing, your neighbor there in New York City. She's had this tremendous expertise in not just pancreatic beta cell differentiation, but this most recently these high content screens. And not everybody can do these, unfortunately. They are expensive, and getting these libraries are a feat in itself. But I think the exciting thing here is this imaging-based modality, this imaging-based screen could be used in the context of other cell types as well. Like, there are certainly markers, visual markers for cardiomyocyte maturation, functional markers for maturation as well, and cardiomyocytes and electrophysiology too. So I can envision this approach, this exact approach, being used for other cell types to find perhaps other cocktails as well.
Yeah, I mean, let's be clear, like the maturation, we're not getting like geriatric neurons here, anything like that, or even to the stage of, I would say, full maturation. I think there's a little bit of work to do to get to that point, but any push forward, I think, is going to be key, especially as the modeling field matures. I think that there's a lot of benefit to be had across the board. But I want to circle back, and I don't know, we don't typically do this on the show, but I would love for somebody listening to the show to come weigh in and tell me how this makes sense, that this paper has been at Nature Biotech, presumably in late review, for like a year and a half. You look at the bioRxiv paper, it is virtually identical. All the results are pretty much the same. There's maybe 1 or 2 panels, additional experiments, calcium imaging maybe that they did to add, but I don't know. I am like vicariously frustrated by the length of time that it took for this paper to get through. And I've had some experience in Nature Biotech myself, like over a decade ago, where the review was way too long. They need some help over there. Come on.
Yeah, it's way too long. I think the review process in general is just becoming way too extended these days. And it hurts the trainees, right? It hurts the first author here. I don't know if the first author here is Emiliano Hergenreiter. I don't know if that's a postdoc or grad student, but regardless, I mean, we need these big publications in a specific time window to advance to the next stage of your career, right? I mean, having a publication like this sit around for 3 to 4 years is just not it's not good for anyone. So I think just I agree with you. I think the review process just needs to be accelerated. And we don't know what happened behind the scenes here. Maybe we can have Shubing or Lorenz on the show to talk more about it. But I agree with you. I think in general, this is just way too long of a process. So we'll shift gears to another Shubing Chen paper. She's also on this particular paper in addition to the last author, Fong Cheng Pan. Over there, once again, Weill Cornell, your neck of the woods. First author here being Zhuohua Duan. And this is another screening-based approach. Again, that's why Shubing has her co-authorship on this particular paper. Cell stem cell paper titled "A Pancreatic Cancer Organoid Platform Identifies an Inhibitor Specific to Mutant KRAS." And perhaps the other reason, of course, Shubing's on this paper is this is a pancreas-related study. Of course, that's her expertise. So we know about KRAS mutations. They're some of the most famous mutations across cancer, and in particular in pancreatic ductal adenocarcinoma, or PDAC, which is this devastating cancer. It's like I think the number 4 leading cause of cancer deaths in the United States, and it's just a really severe prognosis. KRAS mutations are a big cause of these PDAC cases, and in fact, they're found in more than 90% of pancreatic ductal adenocarcinoma cases, and more specifically, a more specific type of KRAS mutation is the G12D and the G12 valine, which are found in more than 90% of PDAC cases. So you need drugs that are targeting these specific mutations, the KRAS G12C or different types of mutations. Some of these drugs have been pretty successful, and this indicates that maybe we can develop more drugs targeting this exact area of KRAS in terms of the amino acid composition. So here what they're doing is, again, a high throughput screen with isogenic mouse pancreatic organoids that are either wild type or actually contain common PDAC driver mutations, including a lot of those KRAS mutations. And these are representing both classical and basal pancreatic ductal adenocarcinoma. So they did, again, big screen, over 6,000 compounds. Actually, in the scale of small molecule compound screens, maybe that's not so big, because some of these libraries can get up to 1 million compounds. So maybe a medium-sized, small-sized screen, I don't know, 6,000 compounds, and identified a particular compound called perhexylene malleate, which can apparently inhibit the growth and induce cell death, specifically of the pancreatic organoids that are carrying the KRAS G12D mutation. And the other cool thing is they demonstrated this perhexylene malleate can function effectively to eliminate the pancreatic cancer cells in vitro and in vivo, and also in primary human PDAC organoids, which is probably why this got to the level of something like cell stem cell. And then they dove into the mechanism as to how this is exactly happening. The single cell RNA-seq, they found that maybe it's the cholesterol synthesis pathway that is being upregulated specifically in the KRAS mutant organoids, including a specific cholesterol synthesis regulator, SREB2, apologies. And so this perhexylene malleate can decrease the SREBB2 levels and reverse that KRAS mutant upregulation of cholesterol synthesis. So that's a kind of a cool tail end mechanistic identification, unraveling of how this particular small molecule and this perhexylene malleate is actually functioning. It's regulating cholesterol synthesis, which is apparently hyper upregulated in the KRAS mutant cells. And again, nice approach to identify a compound that can specifically target pancreatic cancer cells. And once again, and we said this before on the show, this is a severe, dire medical need. We need drugs that can effectively target pancreatic ductal adenocarcinoma. It is a major cause of cancer death here in the United States and around the world. So the work led by Dr. Pan and Dr. Cheng are, this is really critical to helping solve this scourge, right?
Yeah, a scourge with a particularly dreadful prognosis. And it seems to be the one area of cancer where, and I know this is, I'm not an expert here, so I'm speaking out of turn, but the least progress is being made. We talk about, wow, we're curing cancer, but pancreatic, not so much. And I think what we need and what we see here is just a whole new drug. And I think that what's amazing about this approach for me is I love the idea that this model system that was born out of pluripotent stem cells that maybe people thought was far-fetched. And I'm talking about just the organoid system, which existed, I know, and spheroids and all that. And in fact, arose originally with adult stem cells with the famous intestinal organoid. But I just love the idea of how we've kind of circled back into a more traditional drug development paradigm using all of the things that were promised, not all, but many of those tools that were promised. In this case, coming up with with a brand new drug that acts in a really unique way by a unique mechanism, novel, that can be built on either way, either improve the drug, improve like bioavailability and efficacy of the drug in vivo context leading up to clinical trials, presumably, we can hope. Um, but also just this idea of targeting the cholesterol synthesis and trying to come up with other drugs or compounds that follow the same tack. I think that this story has it all for that reason. Not surprising. I mean, Shubing, by the way, put out like 10 papers in the time that it took that one Nature Biotech paper to get in and out of review. So harping on that again, Arun. Sorry, my apologies. But you just can't help it. Anyway, yeah, this is another great story from Shubing. She's Shubing is like my role model and an icon and a rock star to me. And I love everything she does.
And she's just so nice. I've had so many great conversations with her, just such a sweet person to talk to, and that's just the icing on the cake right there. But I agree with you. I think this is a really cool idea, perhaps for, not just de novo compound identification, but and I think that's actually more what they did here was repurposing existing drugs that are out there, right? So this is 6,000 compounds that are known to be out there. I don't think they developed anything de novo, but that's really cool, this idea of repurposing existing drugs, because perhaps in addition to the drugs just being out there, they've already gone through that FDA regulation approval process. So perhaps they can be accelerated towards another clinical application because they've already been validated for their safety, efficacy in other contexts before. So I think a lot of potential for this sort of approach.
Yeah, I'm glad you clarified that because it's not a brand new drug. It's better. It's a brand new application of a drug that's been well vetted. So as you said, so it's like a springboard to clinical application and trial. So I guess, and I hope we won't have to wait too long. I know I'm not alone there. Um, Going from the pancreas to mother's milk here, at least mouse mother's milk. Talking about mammary gland here and the development of more, I would say, comprehensive mammary organoids. I think a lot of organoid spheroid 3D cultures have been generated from mammary tissue, but this is a story on generating mammary organoids that have multiple cell types and function from, in this case, mouse embryonic stem cells. And the key here is like a focus on the ectoderm, right? In the ectodermal lineage, there have been a lot of organoids that have been generated, really sexy organoids. Um, Hair follicle, perhaps one of the lead there in terms of the big splash it made with Carl Kohler at the ISSCR about 3 years ago. Everyone went nuts seeing the hair come out of these organoids, although it was all curly and weird looking. It was still a major watershed, but yeah, inner ear, hair follicle, tooth, organoids, lacrimal organoids, salivary gland organoids. These are all ectodermal derivatives that have been generated, that have had 3D organoids generated from embryonic stem cells. But the mammary lineage, mammary organoids from embryonic stem cells has been more challenging. As I said, organoids have been generated from adult mammary gland or tumors, and those have been really important for understanding the biology of adult stem cells within the mammary gland, and also for modeling like cancerous transformation or the growth and activity of cancer in the case of breast cancer organoids. But of course, this doesn't give us much insight into the developmental biology of the mammary gland and lineage commitment, and all the cell types that comprise the mammary gland. Um, so in this case, the group of Shyam Sharan at National Cancer Institute in Frederick, Maryland, um, kind of played on the developmental similarity amongst the various ectodermal organoids, specifically the fact that mammary glands are thought, I didn't know this, but are thought to have evolved from the hair associated apocrine glands. You know how there's this, the glands associated with the hair follicle can produce all these, sebaceous and all that stuff. So it's thought that the mammary gland evolved from this similar kind of structures. And based on that similarity, the Sharon group um, developed a differentiation approach that was derived from Carl Kohler. And he's an author on this, actually, a couple from the back. Probably helped them with the protocols clearly. And what they did is they adapted that a little bit by using the general protocol, but adding in signaling cues that are known to drive mammary gland development in the embryo. And they came up with this differentiation approach that they call custom, which stands for curating stem cells to generate organoids of mammary lineage. Kind of fits there with the acronym. And they did it. They generated these cells. They did some profiling, did some single RNA-seq to show relative to the hair follicle differentiations that there were more of these mammary cell types. They showed that in Matrigel, they could generate milk proteins in vitro. They had all the cell types, mammary-specific epithelial cells, fibroblasts, and adipocytes, as I said there by the single-cell-seq. They call these things multilineage ESC-derived mammary organoids, another acronym here, MEMOs. And these MEMOs, they underwent ductal morphogenesis, as I said, produced the milk proteins in vitro, and in nude mice, they could reconstitute the mammary gland. They had some really nice pictures showing these TD tomato, which tracked the mammary glands, like really nice arborization and development of the keratins and all that stuff. And then, for a little icing on the cake here, to address the mechanism, which I know they want there at DevCell, they showed that if they knocked out LEF1 and TBX3, which are placode regulators essential for mammary gland formation, they result in impaired differentiation or generation of those memos. So they cover a lot of bases here, I think, with the bottom line being that they're able to generate the constituents of the mammary gland and show that they can function in vitro, which is to say make milk proteins, and that they're able to reconstitute the mammary gland in vivo. And I think it's exciting. I mean, there's a lot of biotechs out there, not a lot, but a few biotechs that are going after making milk from human cells as an alternative to formula. I don't know that this will make that, because, I talked about this a lot with my wife. She's really into, she said, "You're a stem cell scientist. Go start a company where they make milk." I tell her it's not that simple. She's not buying it. But the bottom line, when I looked into it, is that milk from the breast is not just about the casein, the milk proteins. It's all the fats and the hind milk and the fore milk and all that stuff. So I don't know that we're going to have a surrogate for formula just yet. But understanding the developmental process and the cells that comprise this and coming up with a protocol is step one.
Yeah, this is one of the cooler studies that I've seen over the last while, in part because, like what you talk about, that functional validation of these things, right? This actually reminds me of some of the cerebral spinal fluid organoids that Madeline Lancaster, I think, developed. Anytime you can get these organoids to make the things that they're supposed to make, I think that's got a, I guess, a popular science sort of phenotype to it, but it's just also really cool. Those cerebrospinal organoids are making cerebrospinal fluid. That's what they're supposed to make. And these things are making milk. And I agree with you. I think down the road, maybe you can mass produce these things to actually make enough milk ex vivo for whatever application. We're definitely not there yet. I don't think this is going to be translational in any way anytime soon, but it is a really cool technology for nothing, if nothing else, for the basic science of understanding how these developmental processes actually happen. It's not without its own limitation, though. They say it themselves here in the limitation section. These memos, as they call them, have a lot of heterogeneity and also a really big time frame for differentiation. This is one of those 6 month long differentiation protocols, which is, as a trainee, can be pretty frustrating to work with. But, you can't argue with, again, the downstream functional output of these things in terms of the milk production to some extent. But they also allude to that they saw variability in organoid production efficiency from different mouse embryonic stem cell lines. So maybe there's some sort of genetic factors there when it comes to the efficacy of making these things. But nonetheless, I think just a really cool model system that can be built upon.
Absolutely. I just want to clarify, not 6 months. The assay, I guess they do go out to 6 months in some of these, but the assays where they were showing productivity and function was at 30 days. So it's not insane, but I will then circling back to my first Roundup story, that this is mouse. So the time scales for human would likely be much longer. And yeah, as you said, the heterogeneity. This is a approach where they differentiate in a blob, right? And then they, after the fact, they look at the single cell seq and show that all these constituent cell types are there. But it's not, a purified cell type. It's not clean like you would get with these monotypic organoids, like intestinal organoids, for example. It's not as clean. So some limitations there for sure, not least of which being that this isn't a mouse, and we got to see how it works for human, because who cares about freaking mouse breast milk, right? But the extension of this, I don't think, is necessarily going to be a cut and dried clinical application or like industrial application for making milk, which is why it's a dev cell story. And I don't want to set it up like that. I apologize to the authors for making it about milk, because that's not what they're going for at all, to be clear. So yeah, I just want to underscore that this is a developmental study that sheds light on a window that we really haven't had much understanding of, and that's the developmental ontogeny of the mammary gland and constituent cell types. So a big step forward, and that's why you'll find it in DevCell, my friend.
Absolutely. We're big fans of DevCell here on the show, as listeners have heard us harp on for the many recent episodes. We love DevCell. Bunch of great mechanistic developmental stories happening all the time there. So don't forget to check out DevCell. Moving on to another cell family journal, Cell Stem Cell, of course. This is the last Roundup paper for this week. This is, again, a nice basic study with a lot of downstream applications, and it's unraveling the mysteries of reprogramming. And here we are in 2024, Happy New Year, where we're still learning a lot about reprogramming and how it can be made even more efficient. But not just primed reprogramming, but naive reprogramming, which is this new higher order state that I think a lot of stem cell biologists are striving for across different species where iPSC is derived. So this is a paper titled "Highly Cooperative Chimeric Super Socks Induces Naive Pluripotency Across Species." This is I think, a really useful tool developed jointly by Hans Scholler and also Sergei Velchenko. First author here is Caitlin McCarthy over there at the Max Planck, but also some folks at Harvard had a hand in this as well. We're talking about reprogramming, like I said, and we're talking about SOX, SOX2 in particular, which is one of those master regulators of the reprogramming process in combination with OCT4, as we know. And the rationale, I think, is pretty straightforward. We want to make iPSCs and naive iPSCs from a broader range of species. Okay, the iPSC lines that we derive in general, they are artificial. They're an artifact. They're iPSCs, pluripotent stem cells, don't exist in real life, right? But we also just want to make these derivation processes better. Okay. So what they did here is supercharged, I mean, their words, supercharged SOX2, one of the critical transcriptional regulators of the programming process in the pluripotency network. So they actually swapped, they did some really cool protein manipulation here. They swapped some structural elements between SOX2 and an evolutionary analog SOX17 and built this chimeric super SOX, quote, factor SOX217 that ultimately, after validating that this thing is what it's supposed to be, both functionally and at the protein level, ultimately it enhanced iPSC generation in 5 different species that they tested: mouse, human, cynomolgus monkey, cow, and pig. And, mind you, the iPSCs have been derived from all of these species. I think the cool thing that maybe would have taken this paper even further to the next level and beyond is if they were able to derive iPSCs from a species that hasn't had iPSCs derived from before. Okay. I think that would have been really exciting. Maybe they're working towards that. But regardless, diving a little bit deeper into it, they basically did this swap of alanine to valine, a single amino acid change at the interface between SOX2 and actually OCT4, of course, another major regulator of pluripotency. And then this ultimately delivered a gain of function by stabilizing SOX2 and OCT4 dimerization on the DNA itself, enabling the generation of higher quality OSKM iPSCs capable of supporting, and this is important, capable of supporting the development of healthy all iPSC mice. Okay, so you need that validation of the generation of live animals, which is, tetraploid complementation, the validation of one of the definitive validations of IPS production, right? And so the SOX2 OCT4 dimerization actually emerged as a core driver of naive pluripotency, with its levels diminished upon priming, as you might expect. And one other thing that they did was a transient overexpression of an SK cocktail, that's SOX plus KLF4, the super SOX plus KLF4, which restored the dimerization between SOX2 and OCT4 and ultimately boosted the developmental potential of pluripotent stem cells across the 5 species. And ultimately what they want to establish here, or at least, push along, is this idea of having a universal method for deriving naive pluripotent stem cells at high efficiency across mammalian species. This is something I think really, really worth striving for. One of the major limitations of our field, our modern field in general, is just that the iPSCs that we have in general, one, are primed, and two, only from a limited subset of species. I think if I had to take a guess, under 15 mammalian species have had iPSCs derived from them. And maybe this super SOX is something that can perhaps broaden that catalog a little bit more. I mean, we talked about folks like Jean Loring here about the iPSC zoo, right? Being able to derive iPSCs from a huge number of species for conservation processes, conservation applications. And maybe 15 is actually a little low. Maybe it's a little bit higher than that. But nonetheless, I think being able to universally derive iPSCs and naive iPSCs from every mammalian species is something worth striving for.
Absolutely. And I think this is it. My take on this is, first of all, I just want to say, wow, because I wouldn't even know where you start to think about this. I don't know that you're sitting around and being like, oh, yo, I hypothesize. Let's just tweak one of the Yamanaka factors and whatever, and we'll supercharge it. I wouldn't have come at it at all, because I don't know that that's an unmet need, a problem that needs to be solved. But hey, that's why I'm not here in Cell Stem Cell sharing my amazing study with everybody. But I would say, from that standpoint, I think what's underlies that is that we've so many ways to make iPS cells. I think the deep inside, the kid inside me, the scientist inside me is like, wow, this is impressive mechanism. We need this. Now we can make iPS zoo, etc. Easy, industrial, straightforward. But the kid in me is like, I'm not. I've lost interest in another way to make iPS cells. That said, not throwing shade at all, because I want to underscore what I think is the biggest and most impressive thing about this is that it opened the door to this approach of engineering these reprogramming factors. And unlike, induced pluripotency, where there's like literally 100 ways to get to an iPS cell, there's a lot of cell type targets, namely hematopoietic stem cells, anybody, where like we really don't have the recipe. And there've been a lot of approaches that are maybe they're like halfway there, or they get reconstitution, but the cells don't form a lineage or whatever it is. And for me, the real revelation here in terms of concept is, wow, let's try this with some of these hard to achieve cell types where we've been struggling, and maybe we can change the game that way. Also, a good time to note, over the new year, slipped through the cracks, but someone got highly engrafted with hematopoietic stem cells, Arun, from just like EBs. So I'm sorry we didn't get a chance to talk about that, but I bring it up because that's now 40 years on. Combine that with a supercharged engineering approach like this, and maybe we could make a lot of headway towards something that I never thought we'd see the finish line on. So that's my takeaway. Not saying it's boring. Want to be clear. Just saying that I'm not that interested in the IPS aspect of it as much as the engineering aspect and the molecular biology.
Don't be a hater, Dalon, but I agree with you. I think the engineering part of this is really cool. Maybe we think it's cool because we don't do it. But yeah, just being able to modify a single amino acid and getting this supercharged transcription factor as a result, that is applicable, like what you're alluding to, across the board. There's so many master regulatory transcription factors, not just important for pluripotency, reprogramming, but differentiation into blood lineages, cardiac lineages, whatever. And if you can just, if it's, I'm not saying it's easy, but if it's just as simple as modifying a single amino acid to get a supercharged binding in this transcription factor, then I feel like this is something that we should be doing more universally, don't you think?
Oh man, yeah. Not me, but these guys for sure. Because as you said, it can't be easy. That's where I'm like, how do you start? I know it's not, there's ways, but like there's a lot of amino acids to choose from. Why'd you alanine that one up, my man? Anyway, a lot there. I wouldn't know where to start, and Arun thinks it's easy. So clearly that's why we're hosting this show and not doing the work. But maybe we can have Olivier reflect on that in just a minute. Before we get there, have a quick message from STEMCELL Technologies. Do you work with skeletal muscle progenitor cells? The STEMCELL Technologies human myocult workflow supports your muscle research from start to finish, allowing you to derive, expand, and differentiate human skeletal muscle progenitors. You can also expand mouse myogenic progenitors using the mouse myocult expansion medium. Learn more at www.stemcell.com/myocult. All right, everybody, with us on the show today, we have a scientist who's made a major imprint, not only on our understanding of pluripotent stem cells and modeling using those cells, but also just basic developmental biology. Major imprint, a lot of impact from our guest today, Dr. Olivier Pourquié, who is Frank Burr Mallory Professor of Pathology and Professor in the Department of Genetics at Harvard Medical School, also Professor of Pathology at the Brigham and Women's Hospital. Dr. Pourquié's lab is interested in the development of the vertebrate musculoskeletal axis using chicken and mouse embryos as model systems. Members of his lab combine developmental biology and genomic approaches to study patterning and differentiation of the precursors of muscles and vertebrae. Dr. Pourquié, thank you so much for joining us on the show today.
Thank you. Welcome.
Yeah, thanks for being here, Dr. Pourquié. Just side note, it's great seeing you in person. We've had a couple of chats. You may not remember a while back when I was a postdoc in the Seidman lab, just down the road from your lab at HMS Genetics over there in new research building. So I've been watching what your lab has been doing for a few years now. Of course, you've been studying musculoskeletal development, as Delon said, and you've evolved your model systems over the years. And the collaboration that we had briefly was actually in the context of pluripotent stem cells. So it was really cool to see that continue to be fleshed out. Traditionally, as Dalen mentioned, you use the chicken and mouse embryos, of course, standard model systems for studying development. but also more recently, these pluripotent stem cell-based systems. And we'll talk more about the pluripotent stem cell-based models in a little bit, but could you first take us back through the years and tell us what inspired you to actually start working in this area of musculoskeletal development in the first place?
So I started my career in science in the lab of Nicole Ledoirin, who was a very famous developmental biologist who invented the QWERTY camera technique and did very fundamental work on the neural crest development. So this was in Paris a few years ago, and so I did my PhD with her. Ironically, doing mostly molecular biology work, and I was frustrated because this institute was mostly known for the developmental biology work, and I wanted to learn microsurgery and development of the embryo. And there was a professor, an American professor doing a sabbatical, Charlie Ordle at the time, who was studying somite development. And I teamed up with Charlie and we started, so he taught me what he was doing about transplanting somites and so on. And so I became more and more interested in these structures which give rise to the musculoskeletal system. So, and then quickly I bifurcated to segmentation to try to understand the problem of segmentation. But that's how I started in the somite field.
Yeah, the young scientists like Arun and many others may not recognize this, but like the stem cell field was actually created by developmental biologists, and I was about at the I would say, the tail end of that transition from developmental biology to pluripotent stem cells and their tremendous growth as a tool for developmental biology. But yeah, I love hearing that. And I love the arc of your career because it takes me back to those early days where the goal really was to just, take apart human development using other mammalian models to try and figure out what was going on in human development. And pluripotent stem cells, human pluripotent stem cells, were really just like an amazing new tool. And look how far we've come since there. But going back again to that origin there and basic developmental biology, one of your earliest papers out of your own independent lab and the most cited, which is saying something considering the hundreds of high impact publications that you've been a part of. That early paper was a cell story that linked temporal periodicity, aka this molecular clock that you're famous for, to segmentation in vertebrates. Which was really important at the time. It demonstrated that there was a conservation of this mechanism between vertebrates and invertebrates in the fly where it was initially discovered. But, as much as I love developmental biology, I'm relatively naive when it comes to the clock thing. When I hear clock, I think circadian clock, characterization of which led to, was resulting in being awarded a Nobel Prize just a few years back. So I'm betraying myself. Maybe this is a bit of a dumb question, but is the segmentation clock linked to the circadian clock? Is it similar molecular machinery that govern those two fundamental mechanisms of development?
So that's an interesting question. So the short answer is probably not, but that said, in reality, it is a little, and it's just probably by chance because one of the canonical cyclic genes that define the segmentation clock, that is these genes that oscillate in the precursors of the vertebrae and control the rhythmic formation of somites, and this gene is called Has7. It just so happens that the gene next to it is Burr1, which is a critical gene for the regulation of the circadian clock. And so so Burr1 was shown recently to oscillate in the presemitic mesoderm, but that probably doesn't have a real biological meaning. It's just because there is some sort of periodic regulation in the genomic neighborhood, and which also entrains Burr1. But the clock cycle is much faster. It's say in chicken embryo, it's 90 minutes of compared to the 24 hours of the circadian cycle. And actually, if I may just correct something, when you say the clock was conserved between vertebrates and invertebrates, it's only partly true. And in fact, the first gene that we identified in the Cell paper as showing the oscillations was a hairy, so a fly, a vertebrate homologue of a fly payroll gene. But so this was conserved, but it turned out that the global mechanism and this oscillator doesn't exist in flies. It's completely derived. Yet it does exist in other insects, it seems. So a lot of insects or other arthropods appear to have an oscillatory system that controls the production of segments. So, but at the time it was surprising that, you know, the segmentation cascade that you alluded to that was worked out by Nusslein-Bollard and Bichaus, we couldn't find it conserved in vertebrates. And so that's why this oscillator was really came as a surprise in the field, because it was a very different padding from the segmentation cascade of Nusslein-Bollard and Bichaus.
Yeah. Now, fast forwarding a few years, you're also working continuously still on this, segmentation area of study, but using some newer model systems. And we've really loved covering your recent somitogenesis modeling work here on the show, including your recent Nature study of reconstruction and deconstruction of human somitogenesis in vitro using PSCs, pluripotent stem cells, and some really cool reporter systems as well. HES7, I think, was one of the reporters that was in that particular paper. Could you tell us a little bit more about this particular work, using the pluripotent stem cells as the model for somitogenesis, and how these somatoids and segmentoids can help us better understand the earliest stages of human development specifically?
So somitogenesis in humans takes place between 3 and 4 weeks post-fertilization. So it's a time when most women don't even know they're pregnant. So there's virtually nothing which is known, particularly at the molecular level. We don't know whether there is a segmentation clock, if it works like in other mammals, even though there are some hints, like, for instance, the fact that in humans, the very severe malformations of the vertebral column, which are called congenital scoliosis, were shown to be resulting from recessive mutations of genes associated to the segmentation clock, so suggesting that the mechanism might be conserved. But so we've been working on model organisms like chicken and mouse embryos for many years, but then it was becoming difficult to move forward and dissect further the mechanism using genetics, because we just don't have the right precision in terms of spatial temporal control for the experiments. And moreover, it's a very tiny tissue, so it's very difficult to have access to enough material. So indeed, the dream when we started was to be able to set up a system in which we could differentiate cells of the presomitic mesoderm in vitro and recapitulate the normal sequence of events, with the hope maybe to recapitulate even the segmentation clock. And so we did, and in fact, it's work I started quite a while ago when I was at the Star Wars Institute. We started first with mouse embryonic stem cells, then developed protocols based on our understanding of the development of the presomitic mesoderm in the embryo. And then we transferred this to human embryonic stem cells, in fact, to human iPS. And first developed a 2D system with Margarete Diaz-Quadros, a very talented PhD student in the lab, who now has her own lab at MGH. And so what we did was to develop a system where we could image the oscillations of the segmentation clock in 2D. And so Margarete did that, and she identified the oscillator, showed that it had a period of 5 hours, and so on, which was a real feat for us, and was the culmination of many years of work. And now finally we had a 2D system where we could generate as many cells, oscillating cells, as we wanted, and we could start also to manipulate these cells with CRISPR-Cas9, which allowed to generate reporters and mutants much faster than in a mouse. And so after we'd done this work, so I had another outstanding postdoc in the lab, Yuchuan Miao, who started to become interested in developing 3D systems. And he first started by developing a system which was quite unusual in the sense that it was making a 3D aggregates of presomatic mesoderm and then seeded these aggregates in a dish so that they would form a pseudo 3D explant. But what was very remarkable in these explants is that he quickly realized that they were forming epithelial rosettes very much resembling like somites. So in other words, this system was able to make unlimited amounts of human somites in vitro, which was an ideal system to study the process of somitogenesis in humans. So this system has no spatial organization. It's purely temporal. So all the cells in the plate are at the same developmental time, and they develop synchronously. But then he developed a second system, which we call segmentoids, where did a similar approach, forming aggregates of presomatic mesoderm and putting them in matrigel, and which is something that was pioneered by Martinez Arias for the gastruloids. And then this led to an elongation of the structure. And so it really recapitulated the normal anterior-posterior axis, formed somite at one extremity, had neuromesodermal precursors like are found in the tail bud embryos. So it really looked like a freshly micro-dissected presomatic mesoderm from a mouse embryo. This was absolutely spectacular. And especially for it was a bit ironical for me because I spent a chunk of my career, or the beginning of my career, studying the role of the surrounding tissues on the development of the paraxial mesoderm. And here we just drop a ball of stem cells in matrigel, and then it forms, without any surrounding tissue, it forms perfect somites, which are normally polarized. And it's still very mysterious how you can achieve such a level of auto-organization to me, because frankly, it's almost perfect.
Yeah, I mean, I remember I was in the room when Yu Chuan Mao presented that work at the ISSCR in 2022 in San Francisco, and I remember it vividly because I mean, it was a watershed in my career. I had never seen anything like it, um, and no exaggeration, transformative. Because it's, as you alluded to, the degree of spatio-temporally coordinated morphogenesis and the fidelity of it to the real physiological soma. I mean, that's, as you said, it looked like I was watching an explant for an embryo. And perhaps more importantly, as you alluded to in the tail end there, it was this pluripotent stem cell system that was decoupled from, it was organogenesis decoupled from embryogenesis, right? You just said it. It's no surrounding tissue, just this little modular differentiation package you throw in there, and it auto or self-organizes. So for me, that was amazing. And a year later, at this past summer's ISSCR in Boston, blown away again by a similar idea and self-organization with the human embryo modeling, which, you know, people had talked about the mouse embryo, but the human, I think, really made it next level as the segmentoids you showed with using human cells. It's what we've always wanted to do since the beginning, is get a peek into human development, right? And, you know, these studies are only continuing to grow and be applied to gain mechanistic insight. Just in the last show, we talked about 2 stories that use these embryo models to actually ask questions. And I think that's where we're going. But just, you know, in terms of, let's say, next ISSCR 2024, what am I going to be blown away by? You talk about these kind of self-organizing modules, and you were really surprised that there's no role of the extrinsic tissue. It makes me wonder, like, we're able now, the field, is able to assemble these rudiments that then kind of a snowball, like a snowball rolling downhill, can take on the developmental momentum, or Waddington's, you know, cell clusters, or whatever you want to call it, and make organized tissue rudiments at least. What do you think the ceiling on that is? I mean, as someone who's worked in development, you understand that scale is a real issue, right? Without perfusion. But what do you think the ceiling is in terms of us being able to recapitulate, like, I hate to call them mini organs, but like organ enlages that can then develop and give us insight into these primary organogenic events?
Yeah, that's a great question. It's difficult. What currently it seems that one of the big roadblock is definitely the scale up. Because if you look at a human organ, it's big for most of organs, and if you compare to what we do in the dish, it's still ridiculously small compared to. So we're really at the embryonic stage. I think in terms of producing embryonic structures, we'll be able to generate most embryonic structures. I don't see really why this shouldn't be the case. Now, the next phase, the fetal development, where you have this huge amount of growth, something that we haven't really started to deal with. And of course, for translational applications, for instance, it's going to be critical. I mean, you can imagine that one way to deal with is to use surrogate hosts, for instance, because there you probably could achieve much better growth. If you could grow human organs in pigs, or this sort of things, that might be one way to go. But there also, there are constantly some new bioengineering solutions, or which may help driving this growth. The problem clearly is going to be the vascularization. Then you cannot deal with simple. I mean, you have to reconstitute more than one system, in fact, because the nutrient supply is becoming more and more critical as the structure is growing.
Yeah, it's, regardless of some of the potential limitations, in my mind, it's the most exciting area of study in stem cell biology currently. I've said it before on the show, and I'll say it again. If I was a trainee in this field, once again, this is the area of study that I'd be working on. And, by establishing some of these models of somitogenesis and early development, you've also been able to take a unique additional angle on the work that you're doing, and that's looking at the interspecies variation in the rate of embryonic development, sort of more of an evolutionary story that you've put together over the last couple of years. Despite there's a relatively broad conservation of the overall sequence of developmental events. There was this Nature paper that you put out from this year in 2023, where your lab established this in vitro system that can recapitulate the twofold difference in developmental rate between mouse and human embryos. And there's a really profound metabolism story integrated into this particular story as well. So could you tell us more about this recent work, and what it might unveil about how metabolism might influence developmental differences across species?
So this is, I mean, that started. It's a question that I've had in my mind for a long time, because working on clock, you think about time. If you think about somitogenesis, the period of the oscillations of the clock is species specific, and it's plugged into the developmental rate. That is, the species that develop fast have a fast oscillation of the segmentation clock. The species which develop slowly have a slow period of the oscillation. So there's a scaling between these two quantities. And the question of developmental timing is a very fascinating one. I mean, I usually show a picture in seminars which compare in the same dish a 6-week human embryo with a 4-day chicken embryo. And the people I've shown this, the actual dish, before taking the picture, were not able to tell me which was which. And what this says is that you can achieve exactly the same morphogenesis at 37 degrees, so very similar conditions, but at a completely different pace between species. And so what controls this is very, very unclear. And so with Margrethe, we decided to use our in vitro system, the system she developed for human and mouse oscillations in vitro, and use this as a proxy for time to see how we can challenge the system and see what control the regulation of these the period of these oscillations. And so what she found is that the metabolism is a critical element of this regulation, and particularly the energy metabolism in the mitochondria. And what was surprising is that it didn't seem to be directly linked to ATP production, even though it implicated the electron transport chain machinery, but rather the redox status of the cells and more the ratio of NAD over NADH in the mitochondria in the cells. So this was really a surprise and really interesting. And there she did an experiment, which to me was the killer experiment that I wouldn't think would be possible, which was to show that you can accelerate the period of the oscillations, because often slowing down is not enough, because you can make the cell sick and you can find all kinds of criticism. But to try to accelerate the developmental time and hence the clock, and she did so by overexpressing a bacterial enzyme called LB-NOx, which is able to oxidize NADH, so it bypasses the entire electron transport chain. And so in this way, it increases the ratio of NAD and NADH in the cell. And she showed by infecting the oscillating cells with this expressing LB-NOx expressing virus is able to accelerate the clock. I think this was really a very cool experiment, because that's the only one I know of where you have a significant acceleration of the clock. And then she linked it to the regulation of translation, because the very striking thing with this regulation of developmental timing is that it's a global regulation. So another example would be if you take chicken embryos, take them from the incubator, put them on the bench, they will stop developing. If you put them back in the incubator, they will resume developing. If you change the temperature, they will slow down. But it remains, the system remains harmonious, which means that all the chemical reactions that are important for growth and morphogenesis remain coordinated. And that's pretty mind-boggling. But so you have to find about global systems like translation, redox, and metabolism, I think, to explain this.
Yeah, I that story, and I mean the series, I think, of stories that you're telling along those lines were so mind-boggling as you describe it to me, because it's one thing to note and describe a phenomenon that takes place over the course of evolution. We've evolved to have these different time scales across different species, but as you said, that killer experiment where you can actually intervene and change that pace. And I think all of us in the field really recognize the power of that idea, because it shows that the developmental timescales that maybe have been a hindrance in generating the cell types that we want, of the maturation state that we want. While that has been a challenge, it's not an intractable challenge. It may be overcome, and it may be the chemistry and metabolism that allows us to get there. So I think this is another point of evidence. I'm not drawing a direct parallel, like this is the method to get mature heart cells, cardiac cells, or advanced stage neurons or anything, but the concept for me at the time was a big deal, because it showed that it wasn't some static thing. It was something that could be changed. And I think that's an important result. Very deep, pulling back a little bit. I don't want to say superficial, but not as like Evo Devo. I want to shift gears a bit to the story, metabolism related, but only adjacently, about brown fat. You had this STEMCELL paper describing the ontogeny of brown adipose tissue in mice, which was kind of unknown how it came, where it came from, and how. And then built on that to generate this, develop this novel protocol for differentiating human pluripotent stem cells to brown adipocytes. Of course, I mean, if you know anything about brown fat, it's derived from paraxial mesoderm, which has been the focal point of your research for going on 3 decades now. So that it's clear, like, I wouldn't want to call it low-hanging fruit or anything, but it was in your wheelhouse. So I could see why you're looking at brown adipose tissue. But what else motivated this study in terms of rationale, and how do you envision protocols for brown fat differentiation that you develop? How do you envision them being applied, either academically toward basic insight or translationally, perhaps?
Yeah, so there was a little bit of serendipity here, in the sense that Jérôme Chal, who's the postdoc who developed the initial protocols to make paraxial mesoderm and muscle stem cells, observed that by tweaking a bit the conditions of the muscle differentiation, he was seeing some fat cells in the dish. And then we realized that there's been work from Patrick Sear and Bruce Bigerman showing that brown fat had the same origin as the skeletal muscle. So they derive from the same PAX7 expressing precursors. And so we said, oh, that looks really interesting. We should optimize the protocol to try to differentiate more efficiently brown fat, because brown fat is a lineage which is poorly understood, so that its development has not been very well characterized in the embryo. And it's very interesting lineage. It's involved in thermogenesis. So what it does is it's burn glucose and lipids to generate heat. And so, we felt that first improving this protocol and being able to produce large amounts of brown fat would be interesting to better characterize its physiology, but also from a translational standpoint, because as I said, it's very important for the regulation of the metabolism and the glycemia, because it burns glucose. And there is some interesting evidence showing that in humans, the amount of brown fat is correlated to the probability to develop cardiometabolic disease. So some really nice work from Paul Cohen's lab. And so we decided to rationalize a bit more the process, in that we first performed a developmental series, so taking the brown fat region in the mouse embryos at different times, doing single cell RNA sequencing, and then extracting all the signaling cues that were deployed during the differentiation of this lineage using classical machine learning approaches in the field. And then we transposed all this knowledge in vitro and optimized the muscle protocol, now trying to push the cells toward the brown fat fate. And we were successful in doing so. And then we showed that the cells were functional, they were able to produce heat and so on. But what was really striking, which is not yet part of this study, is that we've also been able recently to show with David Piston from WashU, who studied the role of brown fat in the regulation of glycemia and diabetes. So what they've done was to take mouse embryonic brown fat and graft it into mice that were injected with streptozotocin to kill the beta cells, and thus to use this as a model of type 1 diabetes. And what David had shown is that if you do so, what happens is that you can rescue normal glycemia in these mice by grafting brown fat. And these mice have no insulin, so it's a bit mysterious. That reduces the glucagon levels, but how it works in detail is not fully understood. And then we teamed up with David and showed that when grafting organoids of brown fat developed according to our protocol, in these mice, we could also see the same phenotype, a rescue of normal glycemia in the mice. And so where we want to go from there is see if we can develop cell therapy approaches for metabolic disease in humans to transplant brown fat into patients that have diseases like type 2 diabetes, for instance.
Wow.
Yeah, it's a really unique study, and one with some translational potential as well. And side note, the first author of that DevCell paper, Jyoti Rao, was actually the postdoc who I collaborated with a few years ago in the Seidman lab. So that was really cool to see that paper come out. Shifting gears a little bit, you talked about how you have an interest in the translational side of things. And of course, you're in Boston, Cambridge, this area that is the hub of biotech research, not just in the United States, but perhaps for around the world. So many incredible biotech startups there, real translational angle to that area of the world, that area of the country. And I mean, in the spirit of many folks in the HMS Department of Genetics, you've actually recently founded a startup called Somite Therapeutics, which has a tagline of defining or redefining cell therapy with artificial intelligence. So that's very exciting. Honestly, there's not that much info out there about the company since it's so new, but I'm assuming it has something to do with the recent somitogenesis segmentation clock work that we've been talking about here on the show. Could you give us a little sneak peek of what this new endeavor is all about?
Yeah, sure. So the idea is, I mean, I'm convinced that cell therapy is going to be the breakthrough of the 21st century, because we're getting better and better at producing human cells in vitro, and there are many diseases where you need to recreate the cells, or to reintroduce the cells in the patients, because they're gone. I mean, for many degenerative diseases, that's going to be the only way to go. And so the idea here was really to use the somite production as a platform to produce lineages of the musculoskeletal systems for cell therapy. And there, what we want to do is to be able to optimize the protocols using our understanding of the embryo, but also coupling this to artificial intelligence. So we've teamed up with people who are very good at this, like our founder and CEO Mira is coming from this area. He's been involved in he's a serial entrepreneur who's been setting up several AI companies already. We have also Alan Klein, who's also an expert in this studies, and Cliff Tebin, who's the chair of genetics department. So I think we're really a great team when it comes to working on the musculoskeletal system, and it's a lot of fun. And I think it has a lot of there's a lot of potential, because currently there's no one really working in this area in terms of cell therapy. But when you think about it, it covers the orthopedics, production of cartilage, tendons, and bone. Our first focus is going to be Duchenne muscular dystrophy, because we became very good at producing muscle stem cells, and we want to be able to rescue small muscles, like in the hand of adult Duchenne patients who are, basically when they lose these muscles, they lose contact with the world. And so that's tremendously improved their quality of life, and also provide a great proof of concept that will allow us to move forward to larger muscles. And then we also want to develop the brown fat applications that I mentioned, and also working on tendons and other applications.
Yeah, I mean, the scope is really wide, and you could go on and on about, degenerative conditions, that significant unmet need, NIH funding, all that. But as you also alluded to there, in terms of just improving quality of life for literally millions of people, globally, I think the musculoskeletal system, very fertile ground for probably the most surgeries that are done in today could be addressed with cell therapies in the musculoskeletal system when you consider ortho. So, yeah, fertile ground. I'm really looking forward to see what comes out of that. And maybe throw me some stock if you got some laying around. I might be into that. But more than anything, I appreciate you taking this time to chat with us about not only your career, what you're doing now, but what you've done. And for us, it's great to talk to icons like you who've been in it for so long and from the beginning, during that transition, as I talked about, from developmental biology to actual cell therapy, which is where we are now. And as you said, it's the future too. The 21st century is just going to be one therapy after the other, mostly based on cells and genetic engineering. So very exciting time to be alive, still alive, to maybe receive some of these therapies someday. But before we let you go, I have a couple kind of peripheral questions that we like to ask. First, if you were not a scientist, which would be to the detriment to all humankind, if you were not a scientist, what would you be doing with yourself?
Uh, I'm not sure. I mean, when I was a student, I like languages, so I was debating whether enrolling in science or in studying Chinese. I was really interested in the complexity of the language, and particularly the writing. Otherwise, maybe an historian or an archaeologist, which is probably also some sort of scientist. But
Wow, those are pretty far afield. Linguist, historian, and you ended up in science. You made the right call. Finally, before we let you go, what's the best piece of advice that you've been given, either professional or not?
I think to me was an advice from Nicole Ledouara, who was my mentor during my PhD. She said, focus on science, and if this goes well, then everything will go well.
That's great. That's so simple, elegant, and honestly, the best advice that you could give anybody in any career. Focus on the work, right? But in science in particular, I guess there's a lot of ways you can get distracted, not least of which by your own failure and disillusionment, right? So a great piece of advice. I'm glad you passed that along. And again, thanks, Dr. Pourquié. You are a legend, and we really appreciate taking your precious time to share with us and our listeners.
Thanks. That was a pleasure talking to you.
All right, everybody. That brings us to the end of this episode, first of the new year. Hope it's a happy one for all of you. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all of the interview and Roundup papers. You can also reach out to us on X at STEMCELL Podcast or via email at info@stemcellpodcast.com with feedback or to suggest guests.